In this video, I'm going to be talking about all things vision. All things vision. How does our sense of sight work? How does the eye translate light information into neural signals? And some just about everything else related to how we see. Um, as humans, our sight is our primary sense, more so than smell or taste or touch. Um, our sight is what occupies the most part of our brain, right? We have a whole lobe dedicated just for processing visual information. Um, so there's a lot here. So get ready for some a lot of information. So our objectives are to explain the parts of the eye and how it perceives light. We're going to talk about the difference between amplitude, wavelength, and purity. We will ultimately look at optical illusions and how illusions affect and how we can learn about how we see from optical illusions, including learning about things about depth cues like monocular and binocular depth cues. And then we'll eventually talk about color vision and how we see color and other objects and all those kinds of things. So, like I said, there's a lot here. This will probably be broken up into a few videos. So be prepared for that. Also, I should say that we are skipping ahead a little bit. There's a couple sections that are focused solely on sensation as a whole and perception as a whole. Do not read those yet. We will come back to them after we come back from break. So we're kind of doing things a little bit backwards. We're going to kind of go instead of from which is actually some things that we're going to talk about, instead of going from the more general to the specific, we're going to go from the specific back to the general. So we're just going to dive right into vision. So why not? Why wait? No time like the present. So let's go. So we see because of light. The end. Um, Obviously, there's a little bit more to that, but light in this case, now don't, physics besides, we're going to look at light as a wave. I know that it's also a particle, and that's the cool thing about light. I get all that. But from the psychologist's perspective, light is a wave. And in order for us to understand how we see, we need to talk about what light actually is. So really, in general, it's just a form of electromagnetic energy that we happen to be able to see. The electromagnetic energy is very vast, right? We're talking about everything from like X-rays to microwaves to gamma rays. All that stuff is electromagnetic. We happen to be able to see a very small portion of that that's called the visible spectrum because we can see it. And that's how we see. So the light that we perceive, right? Our eyes are just light detectors, for lack of a better explanation. In fact, I'm going to caution you to do this, but if you're not wearing contacts, if you've ever rubbed your eyes so hard that if you do it kind of back and forth a little bit, it'll actually look like you're starting to see some light. That's because you're stimulating your eye, and the only thing that your eye really knows how to do is to understand light. So it takes that movement and translates it into light cues. So wavelength, amplitude are important. The wavelength is the length of a wave between either peaks, valleys, or midpoints from the baseline. It really doesn't matter. They should be the same. So in this case, we have the wavelength as the length between the two peaks of a wave. Amplitude refers to the height from the baseline, either up or down, is the amplitude. Okay, and how we see determines, or the type of wave that is entering our eye determines what we see. So wavelength is associated with color. So the different lengths of the waves produce different colors in our vision. Again, assuming that we're not colorblind. So the shorter wavelengths are the violets and the indigos, and the longer wavelengths are the oranges and the reds. You can see this is a relatively small spectrum, right? We're talking about wavelengths of 400 nanometers to 700 nanometers. Anything outside of that is either infrared or ultraviolet. Hey, look at that. Those words mean things. So depending on the wavelength, that's the color that we see. In terms of brightness, a brighter color has a higher amplitude. A duller color has a lower amplitude. So the wavelength, the length of the wave, is what determines color, and then the brightness of the color is determined by the amplitude. So you can have, a bright green and a dull green are going to have the same wavelength, but they're going to have different amplitudes. There's something that's called purity that we should just mention briefly. This is where you can add light to a different colors, which kind of gray them out. It kind of just like overpowers it a little bit. So you can talk about how saturated a color is in our spectrum. So that's not really in the book. It's just kind of an extra thing to talk about. But when you add monochromatic light, so this is not natural light, but one color light is added to green or red, it makes them less saturated. Again, it just kind of grays them out. Okay, so that's light. Now let's talk about our light detectors, the eye. You need to know all of the parts of the eye. There's really nothing else to it. You're going to need to know how light comes into the eye and how we then understand it in all of the parts. So basically, the eye really has two purposes. Again, all of it is related to light. It provides kind of a location or a house for neural tissue that receives the light in our retina. And it also makes sure that the light gets funneled appropriately to the most important parts of the retina. That's really what the eye does. So we know that a couple parts of the eye, if you remember this from bio or wherever, you have the cornea, which is the outer surface of the eye. It's what goes above kind of your iris and your pupil. That's really just the transparent window that allows the light in. There's a lens, which is a little bit behind the pupil, which bends the light coming in and refracts it. That's why there's the flip. The iris is the color part of your eye, which is actually composed of muscle. We usually don't think of that, but the iris itself is what constricts or expands the pupil, right? The amount of light that's coming in. So in dim situations, when there's not a lot of light, your iris expands the pupil to allow more light into your eye. When it's bright, your iris contracts the pupil so that it's small, so that it doesn't hurt your eye. That's why if you ever go from a dark room to a bright sunny day, your eyes hurt. It's because it's painful. There's too much light coming in. Constricts was the right word. How about that? Okay, so that's kind of the stuff at the front of the eye. We'll get more into the stuff in the back of the eye in a little bit. Okay, so that's kind of a really creepy, frontal view of a disembodied eye, but it works. The things on the sides that kind of make it look not round, those are the muscles that allow us to move the eye, from side to side or up or down, those ones things. Okay. So transduction, we haven't talked about that. We will later. Transduction is to give you the quickest definition possible, is how we take external information and turn it into sensory information. So how do we take something like light and enter that into our nervous system? That's what transduction is, but we'll get into that later. So again, light comes in through the cornea, passes through the cornea, goes through the pupil, which is determined, the size is determined by the lens or by the iris. It hits the lens, it gets refracted so that it goes upside down, and that upside down image then kind of lands on the retina, which is the back part of the eye. And that is where we have our photoreceptors, light receptors, which then translates that information into appropriate neural signals, which then get interpreted by the brain as to our entire visual world. Again, the more you think about those kinds of things, the kind of trippier it gets, or at least it should get. It's very mind confusing. What am I talking about today? All right, retina. So let's talk about this part because this is where it becomes really important. So the retina is really a collection of neural tissue that lines the back inside surface of the eye. It absorbs the light coming in and sends that visual information to the brain through something called the optic nerve. Okay, so if we zoom in, we'll see a number of things. We'll see a number of different types of cells, some of which look like neurons, and some of which, look kind of oddly shaped, weird looking things. So axons, as they leave, these are the light blue at the top of the image. Let's see if I can get my cursor there. Is like here, right? Those are called ganglion cells. The axons of those ganglion cells converge and form the optic nerve. So the optic nerve, which is what allows you to see, is really just a collection of all of your eye's ganglion cells that then transmit that information through one axon, right? It's one neural pathway. That's why vision is so fast. Into our thalamus, which then routes the information to our occipital lobe. Now, you'll notice that when the optic nerve leaves the eye, right? When the optic nerve leaves the eye, there are no photoreceptors there. That's what the red and blue things are along the back wall of the eye. Those are rods and cones, which we'll talk about in just a second. But you'll notice that there are no rods or cones there as all of those axons converge and go out of the eye, which means we actually have a blind spot. Your book has a good demonstration of that, and we'll do one in class as well. I'll have you draw it in your notebook. It's pretty interesting. We don't see it normally, mostly because we have two eyes. So whatever's falling on the blind spot in my right eye is not falling on the blind spot in my left. But even if I close an eye, right, I still looks like I have a full visual field, right? There's not like a big hole in the middle. And we'll talk about why that is. That's because your brain is filling in the gap. Your brain is remembering what was there. And as your eye moves around a little bit, the information then lands on spots that aren't the optic nerve or the optic disc, which then means that your brain remembers what's there and just fills in the gap. So it's kind of a cool little trick. We'll do something in class, a little demonstration related to that, and playing with the way that your brain fills in the gaps. But more on that later. So let's talk about these rods and cones, because this is really where it becomes extremely important. The retina itself is really just two types of cones, these rods and these cones. Classic rods, if you can remember that far back. If you're an old guy, you know what that means. So both have different functions, but both are basically just the basic photoreceptors. They're the ones that are processing the light, the information that comes in. Rods are longer. You can see that in the image, and they're responsible for our vision at night. They are more receptive to dim light than bright light. Cones, on the other hand, are much more prominent in bright lights and color. Cones are what allow us to see color. So you can remember that. It's pretty easy. C for cone, C for color. That kind of means that's an easy way to remember it. Cones are also help us see what we'll call acuity, right? Fine detail. So in parts of the retina that have more cones present, that's where we see things in a finer detail. So that's why something might look a little fuzzy if it's in your peripheral vision, but then you look at it and it becomes clear. That's because of that information is then being processed in the right spot of your retina. So already talked about the optic nerve, already talked about the blind spot, but the fovea is a phrase that you probably might not have even heard of before. I'm assuming that you've at least heard of like the cornea and the iris and the retina, but the fovea is actually the central part of your retina, and there are no rods in that part of the eye. It is only cones, and because it's only cones, and you can even see in this image that it kind of like dips, like all of the ganglion cells and bipolar cells all like kind of branch away there. So it's just cones. It is the most focused part of your eye. If you wear glasses, if you have vision problems or something, what your glasses do is they actually refract the light so that it goes into the lens so that things get projected onto your fovea accurately. What's happening when you take off your glasses is the light's coming in, your lens is misshapen. Sorry to break that to you. Uh, and it's sending the light information to parts of your eye that are parts of your retina that are not your fovea. So things look a little blurry. Um, your book will have more detail about like nearsightedness and farsightedness, but if we're just talking about like more like blurry vision in general, um, regardless of the type, it's because your lens is refracting the light onto your retina, not at your fovea. So your corrective lenses, your glasses or contacts, change the light so that it goes through your lens appropriately into the fovea. That's how that works. I think that's pretty cool. As someone who doesn't wear glasses or contacts, I'm lucky enough for that. But I think that's really interesting how those work. So let's get into rods and cones. So this is the look of your retina, if you will. And we have these rods and these cones. We know that they are photoreceptors. We know that rods are much more sensitive to light. They can't distinguish the different frequencies of color, so they don't really see the wavelengths there. Cones, again, are less sensitive, so that means that they pick up more light, but only respond to light waves of greater amplitude. So that has to be brighter. Right? Again, just think about that. Right? Cones, we see color better in the daytime than we do at night. That's because our cones are more active when there's brighter light. Cones are also a little bit pickier in terms of what light they respond to. So we actually have, again, assuming you're not colorblind here, we actually have different cones for different colors. So there are cones that respond only to blue or other short wavelength light, some that respond to green, some that respond to red. And you can see in this little chart here that rods are somewhere in the middle. But again, cones are what allow us to see in great detail. Cones are what allow us to see color. Rods are what allow us to see, kind of more in dimmer light. And rods are what allow us to see in our peripheral vision. Again, the fovea is where most of the cones are located. You get outside of that, let me go back to this image here. You can see there's not a ton of cones outside of like the dead center of your eye, right? There's more rods in the peripheral of your retina. So that means that your peripheral vision, one, that's why it's a little blurry, right? If I'm looking at things on my desk that are just out of my main visual field, they look blurry until I move my head to them, and then they come into focus. So that's because that information is going into my eye and it's landing on a part of my retina that is dominated by rods instead of cones. Okay. So a little chart for you to use as we go through this. There are significantly more rods in our eye than cones. It's just cones are more centrally located and are responsible for the more detail and color sensitive parts of our vision. Rods, on the other hand, way more of them located on the periphery and are much more sensitive to dim light. So they're responsible for us being able to see at night. They're responsible for us being able to see in our peripheral vision. And we'll do an in-class demo when we come back from break, talking about the how our peripheral vision works and how our color vision works, because I have a pretty cool demonstration to show exactly this. Okay, that'll be it for the anatomy of the eye. We'll come back with another video on visual processing and the pathways in our brain and stuff like that. So stay tuned for that one. See you guys then.